Hi guys and welcome to my channel. Myself Dr. Akshay, and in today's session I'm going to be discussing about the topic diffusion. If you guys want to learn more about the transport of substances through the cell membrane, then click subscribe and the bell icon. So let's do this together. Coming to the topic diffusion. So here in this diffusion there occurs a random movement of particles through the intercellular spaces in the cell membrane, or it can also occur in combination with a carrier protein. So the diffusion through the cell membrane are of two major types. The first one is simple diffusion and the second one is facilitated diffusion. So here in the simple diffusion, the kinetic movement of ions or molecules occurs through the intercellular spaces of the cell membrane without the help of a carrier protein. So here in the simple diffusion, there won't be any interaction with the carrier protein. So here in the simple diffusion, the net rate of diffusion is dependent upon three major factors. So the first one is amount of substances which are available. And the second factor is velocity of kinetic motion of the particles. And the third factor is number and sizes of the openings in the membrane through which these ions or molecules can move. Next, coming to facilitated diffusion. So in this facilitated diffusion, this diffusion process requires an interaction with the carrier protein. So the carrier protein aids in the transport of substances through the cell membrane by binding with them and then shutting off from the membrane through this form. So the simple diffusion can occur through the cell membrane by two different pathways. And the first one is through the interstices of the lipid bilayer. This happens only if the substance is lipid soluble. And the second one is through the watery channels that penetrate all the way through this lipid bilayer membrane. So next, coming to how diffusion of lipid soluble substances occurs through this lipid bilayer membrane. So the lipid solubilities of carbon dioxide, oxygen, nitrogen and alcohol are very high in this lipid bilayer membrane. So these substances dissolve directly in the lipid bilayer membrane and after that dissolves all the way through the cell membrane. And next, coming to aquaporins. So we already know that this water molecule is highly insoluble in the lipid bilayer membrane. And this is because of the distribution of hydrophobic part over the surface of the cell membrane. So in order for the water molecules to pass through this cell membrane, certain protein channels are present all the way through the cell membrane. So many of the body's cell membrane contains protein pores known as aquaporins. And these aquaporins are highly selective for the transport of water molecules very rapidly through the cell membrane. And these aquaporins won't allow any other ions or molecules to pass through these pores. So the total amount of water molecules that diffuses in each direction, that too in each second, is almost as 100 times the total volume of the total RBC cell itself. So there are 13 types of aquaporins that are found in different cells of the human body. So next, coming to how diffusion occurs through these protein pores and channels. So protein pores are distinguished by two important characteristics. So the first one is selective permeability and the second one is gating of channels. So coming to selective permeability of protein channels. So many of the protein channels are highly selective for the transport of specific ions or molecules across the cell membranes. And this selectivity depends upon the characteristics of the channel, such as diameter, shape, nature of electrical charges and chemical bonds with its inside surface. For instance, we can say that this potassium ion channel will allow for the passing of potassium ions almost as 1,000 times greater than the passing of sodium ions through the sodium ion channel. And this is because of the increased size of the potassium ion channel than that of the sodium ion channel. So, see this figure. This figure represents a potassium ion channel. So here you can see this is a tetrameric structure consisting of 4 protein subunits surrounding a central pore. So at the top of the channel you can see are the pore loops that forms a narrow selectivity filter. So lining the selectivity filter are those carbonyl oxygens. So when this hydrated potassium ions enters this selectivity filter, these carbonyl oxygens which are present in this selectivity filter will interact with these hydrated potassium ions. So after this chemical reaction, the shedding of water-bound molecules will occur, and thus leading to the permitting of dehydrated pure potassium ions into the channel. So next, coming to gating of protein channels. So gating of protein channels provides a mean for controlling the ion permeability of the channels. So as you can see in this figure, a strong negative charge on the inside of the membrane can cause these outside sodium gates to remain tightly closed. On the opposite side, when inside of the membrane loses its negative charge, this allows for the sodium ion channels to open its gates on the outside surface, thus allowing for the massive sudden influx of sodium ions towards the inside of the cell membrane. So this process is the basic mechanism for eliciting action potential across the neuron. And in the bottom panel of this figure, you can see these potassium ion gates found in these potassium channels are facing towards the inside of the cell membrane. Here, these potassium gates will open when inside of the cell membrane are positively charged. And these opening of potassium ion channels are responsible for terminating action potential. And the details of this concept, action potential, will be dealt in the further upcoming sessions. And some protein channels are opened by binding of a chemical substance called ligand with the protein. And this causes a conformational change in the protein molecule that further opens or closes the gate. So one such example of chemical gating is the effect of acetylcholine over the acetylcholine channel. This acetylcholine opens the gate for this acetylcholine channel, thus providing a negatively charged pore of 0.65 nanometer diameter, and this allows for the passing of neutral molecules or positively charged ions through this channel. And this gating is very important for the transmission of nerve impulses from one neuron to next neuron and from one neuron to the muscle fiber, and thereby helping in the muscle contraction. So that covers the topic diffusion. I hope you guys learned about this particular topic. Thank you so much guys. See you guys in the next session.